Bismillahirrahmanirrahim. We start another video on the June 2024 paper, and this is now the paper 2-1. Bismillahirrahmanirrahim. This is for 1 hour and 45 minutes. Please remember this paper is not for 2 hours, and this is for 1 hour and 45 minutes. Now, coming to question number 1, the lungs are part of the human gas exchange system. Breathing moves air into and out of the lungs. Figure 1.1 shows parts of the human gas exchange system and their functions. On figure 1.1, draw a line from each part to its function. One has already been done for you. So the intercostal muscles, what are they going to do? They're going to change the volume of the thorax. So this goes here. Then it says goblet cells. What do goblet cells do? They release mucus. So this goes here. Then alveoli has been done for you. Then we have cilia. What do the cilia do? Cilia remove pathogens. So you connect this. Then what does the trachea do? Trachea carries air to the bronchi. So this goes here. So you just have to draw lines, and this was for 4 marks. So if you got 4 correct, 4 marks. If you got 3 correct, 3 marks. 2 correct, 2 marks. 1 correct, 1 mark. Then it says state 2 features of alveoli make them suitable for gas exchange. So if you please be careful when you're writing it, because if you write 2 incorrect ones in the beginning and then 2 correct ones at the end, so it will be +2 -2. So be very careful what you write and how you write it. Even if you in the first line write 2 points, and these 2 points are wrong, and then you've written 2 correct points, well, we're not going to give you any marks for this. Now, you see there are many alveoli. Now, of course, all these many alveoli is going to increase the surface area, so large surface area. Then there are the walls of the alveoli are made of very thin cells or flattened cells. And then inside the alveoli, you have a layer of water. And this layer of water, of course, helps in the oxygen dissolving and moving into the blood. And then, of course, the other thing is that it is very close to the capillaries. The capillaries are very close by. So basically, oxygen has to diffuse from here into the alveoli into the blood and then combine with hemoglobin inside the red blood cell to form oxyhemoglobin. So large surface area, thin flattened cells which make up the wall of the alveoli, and then there's a layer of water, or you can say a layer of moisture. And the fact that these alveoli are very close to the capillaries, these are the capillaries which are found just very close by. And the capillaries are also 1 cell thick, but of course we don't talk about that because they've asked you features of alveoli that make them suitable for gas exchange. And coming to the B part of the question, it says gas exchange changes the percentage of some gases in the lungs. The pie charts in figure 1.2, here is the pie chart, this is a pie chart, and this is a pie chart, shows the percentage of gases in the lung change during one breath. Complete the pie chart titles. So what will be the title of this? This will be inhaled air, and this will be exhaled air. Because you can see something is, this was more, so this has to be oxygen, this was 20%, now it is 16%. And then they have given you a table, and then they say, okay, what do you do? Complete the pie chart and the key which shows which gases are represented. So what is represented by this? This is what? This is nitrogen. 78% is nitrogen. So this is nitrogen. Then what was this? That was oxygen. 20% oxygen and then 16% oxygen. And then of course they've given you this as other gases. So what is the black part, which is of course more in it? So that has to be carbon dioxide. Because there's very little carbon dioxide in inhaled air. So this completes this question. Then it says question number 2, brown bread made from the flour of whole wheat grains contains both starch and fiber. A man eats a piece of brown bread. In his digestive system, the starch in the bread is chemically digested to maltose and then to glucose. Describe where and how the digestion takes place as the bread travels through the digestive system. So amylase acts on the starch. Amylase is in the mouth and in the small intestine, or you can say the duodenum. Then the maltase acts on the maltose and produces glucose. Maltase is found in the small intestine, or you can say even the duodenum or the ileum. All was accepted. So 4 marks, and there are 4 mark scheme points. Amylase acts on the starch. So you're giving me what? It says how this digestion takes place. So amylase acts on the starch, and amylase breaks down the starch to maltose. Where is the amylase found? It's found in the mouth and is found in the small intestine. Then you have another enzyme called maltase, which acts on the maltose and results in the breaking down of the maltose, which is a disaccharide, into glucose, which is a monosaccharide. And where is the maltase found? Is found in the small intestine. So basically 4 marks, 4 mark scheme points. So this is how we'll word it. Amylase acts on starch, produces maltose. Amylase found in the mouth or the small intestine. Maltase acts on the maltose, producing glucose, and the maltase is found in the small intestine. Then coming to the part 2, describe what happens to the fiber as it passes through the digestive system. Very simple, very basic. It's not digested, or if you said it's not broken down, is passed out in the feces, or you can say is egested out of the anus in the feces. So basically, fiber, why can it not be digested? Because we don't have the enzyme cellulase. We do not produce the enzyme cellulase. So we cannot digest the fiber. So fiber is going to pass out undigested. Then coming to the B part of the question, glucose passes from the digestive system into the blood by diffusion and active transport. Give 2 ways in which active transport is different from diffusion. So active transport requires energy. Active transport moves molecule against the concentration gradient, and active transport requires carrier proteins. But any two of these, and you would get your 2 out of 2. So basically, because it talked of active transport first, so then you will talk of active transport. Otherwise, you can do a comparison and say, okay, diffusion is different. Diffusion doesn't require energy. Active transport requires energy. Or you could have said active transport requires ATP. ATP is a universal energy currency. The glucose has to be converted to ATP so the cells can use the ATP. We cannot use the glucose in our body cells. Our body cells can only use ATP. Like your car runs on petrol, so you're not going to put diesel in it. So our body runs on glucose, but the glucose has to be converted to ATP because ATP is the energy currency used by the cells. So this is how we're going to word it. Active transport requires energy. Active transport moves molecules against a concentration gradient, or you could have said moves them from a low concentration to a higher concentration. And active transport requires a special carrier protein, which is different for every different ion which is being actively transported. C part of the question says scientists have found that there are different types of starch which are molecules of different shapes. Some molecules are digested rapidly and others are digested slowly. The graph in figure 2.1 shows the effect on the concentration of glucose in the blood after eating 2 types of starch. There's a rapidly digested starch, which is this one, and you can see it increases. What is it increasing? It's increasing the blood glucose concentration and then it's decreasing. And the slowly digested starch, it is increasing, but then there's not such a higher level of glucose. It not really goes up a lot and then it decreases. And it stays a little higher than the rapidly digested starch. Now, it says there's already glucose in the blood when the starch is eaten. Explain how figure 2.1 shows that. Well, because it's starting from here, the blood glucose level. This is the zero. Zero is here. So it must be, say, 50 here, and then 100, and then 150. Whenever you're doing a graph, please always put some figures on the y-axis and the x-axis. X-axis, of course, has figures here in this situation. But in A levels also, I tell you that, always put figures on the graph. So it was already 50 or 100 or whatever it was. So there is already glucose in the blood. It says, but how does figure 2.1 show this? It shows this because the graph line does not start at 0. The graph line, if you look at it, I'm going to just erase all this. The graph line starts here. It doesn't start at 0. So the graph line does not start at the origin. The graph line starts partway up the y-axis. Then it says both graph lines show an initial increase in blood glucose concentration followed by a decrease. Suggest two reasons why the glucose concentration decreases. Now we all know that. Why? Because glucose is being converted to glycogen because insulin has been released, or the glucose is being used in respiration for the release of energy because it's your body fuel. It's just like you put petrol in the car, and by the evening the petrol has been used up because you've been traveling to school and back, and then so the car is being used. So suggest two reasons why the blood glucose concentration has decreases. Number one, the glucose is converted to glycogen because high blood glucose level result in the release of insulin. We didn't say produce insulin, we say insulin released. There's a large difference. And the glucose is being respired to release energy. So graph line does not start at origin, then glucose converted to glycogen, insulin released, and glucose respired to release energy. Then part 3 says using information from figure 2.1 to suggest which type of starch is healthier to have in the diet, give a reason for your answer. Now, if you look at the graph, what has happened? The rapidly digesting starch results in a lot of glucose spiking. So the glucose level went up a lot. But the slowly digesting starch, it did not go up a lot. It just went up to here, then down. So there's less variation in blood glucose concentration. If we took slowly digested starch, is going to result in less variation in the blood glucose concentration, and the pancreas may not have to produce so much insulin. This may be helpful because it is less likely to develop diabetes. Because what is diabetes? Diabetes is because your intake of carbohydrates has been a lot, and because, as I said, the intake of carbohydrates has been excessive, so that has resulted in high blood glucose levels, and so the pancreas has been producing a lot of insulin. Now, one of the theories, I'm not sure it's been proved, that people who take a lot of carbohydrates finally develop diabetes because the pancreas sort of, gives way and doesn't want to make any more insulin. It's tired, or maybe it's just packing up. So that is why in this question, what we had to say was using the information suggests which type of starch is healthier. So the slowly digesting starch is healthier, but that doesn't get you any mark if you don't give me a good enough explanation. Just writing slowly digesting starch is not going to get you any marks. Less variation in blood glucose concentration is going to get you a mark. Pancreas not having to produce so much insulin will get you a mark, and less likely to develop diabetes will get you a mark. So, slowly digesting starch, of course, you've given that as the heading. So, less variation in blood glucose, pancreas not having to produce much insulin, and less likely to develop diabetes. Part D of the question is scientists want to produce a new wheat variety with an increased fiber content. Describe two methods they could use to do this. Artificial selection, you select plants and then with a greater fiber content, and you breed them, take the pollen, deposit it on the stigma, then wait for the seeds to develop, and you repeat this over many generations. Or you can do genetic modification. You can put in a certain allele, you can even use the word gene, an allele from another plant which produces high fiber. So you can either do a genetic modification or you can do selective breeding. So both these would be possible. Your answer would be correct if you gave me any of those 2 points. So these are two different answers, and these two would get you your 2 marks, and then these two would get you your 2 marks. So this is 4 marks. So you have to give me, it says describe two methods. So describe means you'll have to tell me what is the method, the name of the method, and then how are you going to do this method. So it's number one is artificial selection, or you can say selective breeding is the same word. So you select plants and then you breed them. Select plants with the greatest fiber content and then you breed them. Select/breed plants with greatest fiber content. And the second is genetic modification, and they transfer the allele or the gene so that the plant produces high fiber. Question number 3, figure 3.1 is a photomicrograph showing part of a capillary network of a woman. The structure labeled X is a red blood cell. The diameter of cell X in the photomicrograph is 10 millimeter. Calculate the actual diameter of cell X and show your working and express your answer in micrometer. And this has been magnified 1250 times. So it's 10 millimeter. Now please remember, always write, we are talking about millimeters. So 10 divided by 1250, right? This will give you what? 0.008 what? You're talking a millimeter. But then it said you have to, 1 millimeter is equal to 1000 micrometer. So you multiply this by 1000 and the answer is what? The answer is 8 micrometer. So 10 over 1250, why? Because the magnification was 1250. So 10 millimeter they have told you in the question. So 10 divided by 1250, this is 0.008 millimeter. And you multiply this by 1000. Why? Because 1 millimeter is equal to 1000 micrometer. So this will be 8 micrometer. Now coming to part 2. W and X are both red blood cells. Explain why W and X appear different from each other in figure 3.1. It's just the view. I mean, if you look at your phone from the side or if you look at it from the top, it's very different. Both have the same shape, but W is the side view and X is a view from above, seen, or you can see it seen from different angles. So both the same shape, W is the side view, or you can say X is the view from above, or you can have said seen from different angles. Now coming to part 3 of the question, Y and Z are both liquids. Name the liquids Y and Z. Explain how liquid Z forms liquid Y. Now let's look at Y and Z. So Y is inside the capillary, so Y has to be the plasma, and Z is outside the capillary, so that is the tissue fluid. You see, they've shown you the capillary. They said this is a capillary network, and then you can see the red blood. So this has to be a capillary in which the red blood cells are traveling. Now they've asked you what is Y and what is Z. Now Y is inside, so Y has to be the plasma, and Z is outside, so that has to be the tissue fluid. And plasma is under pressure, so the plasma passes out of the capillaries to form the tissue fluid. Plasma is inside. Till it's inside is called the plasma. When it's outside is called the tissue fluid. The same liquid which was inside, which is called the plasma, now as it is squeezed out under pressure, the water plus all the glucose and amino acids and stuff in it, that is now going to be called the tissue fluid. So plasma, tissue fluid, plasma under pressure, it is squeezed out because you see the capillaries arise from the arteriole, and arteries carry blood at a high pressure because all arteries arise from the aorta, and aorta arises from the left ventricle, and the wall of the left ventricle is thicker, so it exerts a lot of force. So there's more pressure in the arteries. So plasma is under pressure, and that is going to result in the plasma passing out of the capillaries to form the tissue fluid. B part of the question, the woman has malaria and symptoms of anemia. The malarial pathogen infect red blood cells where they multiply to form many new genetically identical pathogen cells. These new pathogen cells burst out of the red blood cell. Describe how malarial pathogens can be transmitted from this woman to another person. Now that's very simple. What is the main thing? The mosquito is a vector, is going to suck up the blood from the infected woman, which of course contains the plasmodium or the pathogen. And then this mosquito is going to go and suck the blood of another person, and that's when it injects the pathogen into the other person. It doesn't inject it, in fact, it's just passed out in the saliva. The saliva contains the pathogen, and it is a reflex of the mosquito to actually inject the saliva before it takes the blood, because the saliva contains an anticoagulant. But they don't expect you to give me all that detail, but I expect you to understand all that detail. So you just come up with the basics. Mosquito vector sucks up the blood from infected woman, and then this pathogen is transferred to another person when it sucks the blood of that person. So mosquito vector sucks up blood from infected woman which contains pathogen and then injects this pathogen into a different person. Then we come to name the type of nuclear division that results in new pathogen cells. Now you know Plasmodium is a protoctis, it's a eukaryote. So the type of division is going to be mitosis. Now only in bacteria it is not mitosis. Bacteria divide by binary fission. So you have to be very careful where you write mitosis and where you write binary fission. Suggest why infection with a malarial pathogen can cause anemia. Why? Because red blood cells are destroyed, so it reduces the number of red blood cells. Then state one other cause of anemia. There's iron deficiency anemia. If you don't take enough iron in your diet, or there is heavy loss of blood through injury, or if it's a woman and she is menstruating a lot and there's a lot of loss of blood every month, that can cause anemia as well. So type of nuclear division is mitosis, then infection cause anemia, red blood cells destroyed, reduces the number of red blood cells, and cause of anemia is iron deficiency or heavy loss of blood through injury or menstruation. Any one of these, because this is only for 1 mark. We will continue this paper in a second video to finish questions 4, 5, and 6.